speaker number 6 is ChanHee, who's a PhD candidate in our program working in the lab of Dr. Jamie McLeod. ChanHee is from South Korea and majored in swine nutrition for her Bachelor of Science degree. She was motivated by a desire to focus on equine science, thus she came here, and earned her master's degree in the Department of Animal and Food Sciences working with Dr. Christine Urschel. She's now in her 4th year of her PhD program. ChanHee.
An institution consists of different departments but works as one organized group. For example, the University of Kentucky is an educational institution. While its major goal is to provide higher education, it is composed of many different departments offering diverse programs. Similarly, a tissue also works as one functional unit, and it is composed of different subunits. And in this case, the subunits are different cell types, and they determine the properties of a tissue. In my dissertation project, I'm studying 2 developmental cartilaginous tissues and their cell populations. One is interzone tissue, which will develop into joint tissue, joint articular cartilage tissue, and the other one is amalgam tissue, will eventually develop into bone. The reason why we are so interested in these 2 tissues and their cell population is that bone injuries generally heal well, while articular cartilage defects do not repair. Thus, understanding comparative aspect of these 2 tissue types and their cell population should provide important information for improving articular cartilage regenerative treatment. To study these tissues, we grew them in 3-dimensional cell pellet cultures, and we will evaluate how the cell pellets are organized and what genes they express. Gene expression experiment can be designed either to make measurement at a whole cell pellet level or to measure individual cell populations. Now, let's look at the analogy illustrated on this slide. On the left side, we can see a broader perspective. Like going on a general campus tour, the traditional bulk RNA sequencing will evaluate gene expression characteristic of the whole aggregated cell pellet. Now, let's focus on the right side. Like visiting each individual college or academic department, the single-cell RNA sequencing will interrogate individual cell and profile component cell population of a cell pellet. Thus, by studying these 2 tissue types at a single cell level, we hope to learn new knowledge that will have some relevance for therapeutic approaches to help articular cartilage repair. Thank you.